All right, so this is the second video for catching people up. It's basically going to talk about the next little section that we covered. So we did some Punnett squares, and then we did a few questions, problems, and we did the little labs. Okay, so the first part, we didn't really go over uh, this slide per se. It's there for you to refer back to when you're trying to do Punnett squares, just for little directions, but we went ahead and we looked at some problems. So if you look at the problem and kind of ignore, let's see if I'll put a shade up. Okay, look at the problem itself, and then we'll kind of look at the Punnett squares. It says, create a Punnett square for a cross between a heterozygous round and a wrinkled pea plant. Determine the genotype and phenotype for each offspring. Then determine the Mendelian ratios for each. So basically, when you're doing these, the key thing is look first and figure out what are the alleles, possible genotype of the parents that you're crossing. Okay, so it says between a heterozygous round. So heterozygous means it has to have two different alleles or two different versions of the gene. Heterozygous round means that it's showing the dominant round trait. Okay, so it'd be big R, little r. And the other parent is wrinkled. So if it's wrinkled, then that's the recessive trait. They must be little r, little r. Okay, so we're supposed to create a Punnett square for a cross between these parents. So when you're doing the Punnett square, you usually write the parent cross. Okay, so you have big R, little r by little r, little r. And then you put the parent genotypes along the sides. So here's here, use this one. Okay, so here's one parent, and here's the other parent. Okay, and then you say what their um, possible alleles they can give. So this one can give a big R, or it can give a little r. This one can only give little r's. Okay, so you show what possible gametes they can, what possible alleles they can give in the gametes. Okay, inside the boxes of the Punnett square, you combine the two letters. So if this one gives a big R, and this one gives little r, then the gametes together would be big R, little r. And that one, that genotype, big R, little r, would appear or have a phenotype that is round. Okay, so sometimes it helps if you write the phenotype right in the box too. Okay, so this one would also be big R, little r. This one gives a little r and a little r. So you go take it from the row and the column, what letters meet. Okay, and this would be little r, little r. So the phenotypic ratio is what you see. So we said there's two round, and the bottom ones are two wrinkled. So two round to two wrinkled. You could also say one round to one wrinkled, right? Because 2 to 2 is the same ratio as 1 to 1. Oftentimes it's easier to look at your total numbers. So if we were looking at 4 offspring here, then you'd have 2 to 2. Okay? If we're talking, if they said say percent, you could say 50% are round, 50% are wrinkled. The genotypic ratio, remember, is the letters. So we have 2 big R, little r's to 2 little r, little r's. Oops. Now, I'm going to get rid of that. Get rid of that. Okay, so I assigned a few problems at that point for them to try. I wanted them to do number 3, number 10, number 11, number 13, and number 14. Okay, so then we went through and we did another sample one. Okay, I said, let's take flower color. Oops, flower color. And I want you to show me the F1 and F2 generations you get when you cross a purebred purple and a purebred white flower. So because we're doing purebred purple and purebred white, and this is a particular cross that Mendel would have done, you're going to end up seeing the same results that we saw when we looked at his crosses. Okay, so in the F1 generation, because we have, let's see if I can change the color of the pen. Oops. Everything's the same color. Okay, so because we have big P, purebred purple, versus purebred white, or little p, okay, this parent can only give big P's, and this parent can only give little p's. So when they're combined, all the babies are big P, little p. Okay, all the F1 offspring are big P, little p. So that means that they are all, or 100%, will be purple. That's what you see. Okay, as a Mendelian ratio, you'd probably say 4 purple to 0 white. Okay, that would be, because it's what you see, that's the phenotypic ratio. The genotypic would be 100% big P, little p, or 4 big P, little p to 0 big P, and 0 little p, little p. Okay, in the F2 generation, so if we took these F1 babies and we cross them by themselves or self-fertilize them, or in other words, because you're crossing them with themselves, they're going to have the same genotype, big P, little p by big P, little p. Okay, here's the parents. So the F1 by the F1, big P, little p. Each can give a big P and, oops, a big P and a little p for the gametes. Okay, so this one can give a big P and a little p for possible alleles in the gametes. So when you combine them, you have a big P from here and a big P from this one, gives you big P in the offspring. If you have a big P from this one and a little p from this one, we get big P, little p. Down here, little p and a big P from the top, we get another big P, little p. And if we take the little p from here and a little p from here, in this box we get little p, little p. So phenotypic ratio, what you see, these three, oops, it's getting kind of messy. These three all have a big P. So they'd all show the dominant purple flower. So it'd be 3 purple to 1, that is little p, little p. Okay, so 1 white. Genotypic is 1 big P to 2 big P, little p's to 1 little p, little p. Okay, so gotta make sure you can tell the difference between phenotypic and genotypic. So they worked on the problems that were assigned. Okay. Number 3 said in cattle, horns are recessive over hornlessness. So if they're hornless, then they're showing the dominant trait, big H. If 2 homozygous cattle, one hornless and the other horned, are crossed, what are the genotypes and phenotypes of the first generation? Okay, so we have 2 homozygous, so that means that they are both pure for whichever trait. One is hornless, so hornless is the dominant trait, big H, and the other is horned, which is recessive, so little h, little h. So there's our parents. That's the first thing we figure out. What is their genotypes? Okay, so here's the crosses. We have one parent here, another parent here. This parent can only give big H's for possible gamete alleles. This one can only give little h's. So you put their gametes along the edges and then you fill in the Punnett square. So we have only possibilities are big H, little h for all of them. So since hornlessness or hornless is the dominant trait, all of these are going to be hornless. And they all show heterozygous dominant alleles. Okay, so big H, little h. That's number 3. The next one was number 10. Oops. Okay, so number 10 said in pea plants, yellow peas are dominant over green. Use a Punnett square to predict the phenotypes and genotypes of the offspring from a cross between a plant that's heterozygous for yellow, so big Y, little y, and a plant that's homozygous for green, little y, little y. Okay, so this is what the Punnett square looks like. Big Y, little y, little y. You know, it'd be really good if you, when you get to this point in the video, just pause it. Try the problem yourself and then look at the answer. Okay? Because there's no sense in me just explaining the answers to you. If you pause it, try it yourself, and then look at the answer, that'd be better. Okay? Because that's what they had done. So we have a big Y from here and a little y from here. Okay, big Y, little y. Another big Y over here and a little y from the top. We get a big Y, little y. And then the rest are just little y's. Okay, so the top two are heterozygous dominant, so they're both yellow. And the bottom two are homozygous recessive, so they're both green. So genotypic ratio, you could say it as 0 big Y to 2 big Y little y to 2 little y, or you could just say 2 big Y, 2 big Y little y to 2 little y. You don't have to put this part because there's none of them. Phenotypic ratio, you'd say there's 2 yellow to 2 green, or you could say 1 to 1, as long as the ratio is held constant. Okay. The next one was number 11. Again, if you go through and try the question yourself first, and then we'll look at the answer, it would probably be more beneficial to you. Okay? So it says, as a reward for being a good student, Mr. Singh gives you a rabbit named Bud. Bud has long hair. You introduce Bud to another rabbit, or your other rabbit, Sarah, who has short hair. Later that year, they produce a litter. Imagine a boy rabbit and a girl rabbit getting together. Anyway, they produce a litter that has 1 long hair and 7 short-haired bunnies. If short hair is due to a dominant gene, okay, and long hair is recessive, what are the possible genotypes of Bud and Sarah? All right, well, let's see. It says Bud has long hair. So if long hair, it says, is recessive, Bud must be little s, little s. Does that make sense? Yeah, okay. So Sarah is short hair. Sarah has short hair, but let's see. In their offspring, they're able to have at least 1 long-haired baby. So in order for them to have a long-haired baby, both parents would have to give a little s. So that means that while we think Sarah could be big S, or big S, little s, in order to have a long-haired baby, she has to have a little s to give that baby, to make it long-haired. Okay, so that's the answer for the first part. And I think, yeah. Okay, so then it says, what phenotypic ratio would you expect in the offspring generation of a cross between Bud and Sarah? Create a Punnett square to show the results. So Sarah would be big S, little s, and Bud would be little s, little s. So we're going to set up the Punnett square. Okay, so there's Sarah's and there's Bud's, and we look at the possible gamete, what's in each possible gamete, which allele, and then you fill in your Punnett square. So we have a big S and a little s, because Bud can only give little s's, right? A little s and a little s, a big S and little s, and a little s and little s. Okay, what phenotypic ratio would you expect? So it's only asking for the phenotypic ratio. Phenotypic ratio is what you see, remember, the physical appearance. So we have two of them have a big S. So two of them would be short hair, which is dominant, and two of them have little s, little s, little s, little s. So normally you say the dominant one first, but it's fine. Two long-haired to two short-haired. Oops, goodness, touchy screen. Two long-haired to two short-haired. Or you could say one to one. That means the same thing. How many of the 8 bunnies were expected to be long hair? Well, if you look, 2 of the 4 in the Punnett square, so in other words, half of the offspring, okay, 2 over 4 are expected to be long hair. So if half of them are expected to be long hair, how many of the 8 bunnies would be expected to have long hair? What's half? In other words, you're trying to find half of 8. That would be what's expected. So anytime we do a Punnett square, it's giving us expected results, not necessarily what you're going to see in nature, because nature is by chance. If we looked at a whole lot of different litters that they had, we'd probably get close to what's expected. But this one is by chance. So half of 8 would be 4. You'd expect 4 of them to be long-haired. The expected phenotype ratio would be achieved in every case. Would you agree with this statement? Why or why not? And that's exactly what we just said. Okay, no. I would not agree with that. Okay, it's not going to be, you're not going to get what's expected mathematically every time. That's what we mean when we talk about expected versus observed or actual results. In nature, things happen by chance. The more times we look at the offspring, like if we looked at their litters over time, a whole lot of different litters, we'd probably see that if we counted them all up, then about half of their offspring would be long-haired and half would be short-haired. But for any one particular litter, it's probably not going to be what's expected mathematically. Okay, so that's number 11. Number 13. All right. Again, try and do the question first and then look at the answers. Okay, so in this question, oops, why did I do that? It says, in humans, albinism or lack of skin pigmentation is governed by a recessive allele, little a, and normal pigmentation is governed by the dominant one, big A. Given this information, determine the genotypic and phenotypic ratios of the children expected from the following crosses. So they give a series of crosses, and you have to figure out what the genotypes are for the parents, and then figure out the Punnett squares and the ratios. So I'm not going to go through all of this one. Okay, basically you look and it says homozygous dominant, that means big A, big A. Heterozygous means big A, little a. You have one of each allele. So you set up your Punnett square and you fill in the Punnett square and then you give the genotype and the phenotype, the ratios. Okay, so we've gone through and done all those. Now the questions that you may be wondering about are the one further down. Okay, so these A, B, C, and D are just, basically figure out what they mean by the terms. What are the parent alleles? Do your Punnett square, figure out the ratios. Double check and see that you have the same ones as I got, as we did together, okay, when we checked them. Now, it says, E, for each cross above, state the likelihood of the first child being albino. Okay, so you go back and you look at the results. In A, are there any of them that show the albino trait? Any of the offspring in the Punnett square? So you look at your phenotypic ratios, none of them show the albino trait, so you'd say the percent likelihood would be 0. Okay, in B, the percent likelihood of the first child being albino is 50%. Okay, because half of the offspring, where's my pen? Half of the offspring in B are albino and half are normal. So you'd have a 50% chance of the first child being albino. In C, again, none of them are albino, so you'd have a 0% chance. In D, only one of the four, so 1/4, or if you're trying to figure out percent, you'd have quarter times 100, which is 25%, right? 25% chance of being albino. That's for the first. So then it says determine the percent likelihood of having one normal child followed by 2 albino children in one family. Okay, so again, because there are no chances of having albino children in A or C, okay, you'd be multiplying by a 0. So when we do this, we're doing what's called the product rule. Okay, so if we looked at um, B, okay, and we said it says one normal child. So what's the probability of having a normal child in B? 50%, or 2 out of 4, or a half. Okay, so you could write it any which way, but it's easiest when you're doing the product rule, it's easiest to do in fractions. So one normal child would be half, or 2 of the 4, okay, followed by 2 albino children. So in order for it to be all 3 of these things, we go half for normal times the probability of the next one being albino, which is half, times the probability of the next one being albino, which is half. So you'd have 1 by 1 by 1, 1 on the top, 2 by 2 by 2, 8. Okay, so the percent likelihood of having one normal followed by 2 albino children in the family for B is 1 in 8. If we looked at D, the probability of having a normal child is 3 over 4. Okay, probability of having an albino one is 1 over 4. So you'd have if you're talking about this problem down here, for D, it'd be 3/4 for a normal child by 1/4 for the first albino, and then 1/4 for the other albino. So you'd have 3 times 1 times 1 on the top, so 3, and 4 times 4 times 4 on the bottom is 64. So the likelihood of having that is 3 over 64. Okay, so that's the tricky part about number 13. Number 14. All right, this one is kind of a process of elimination. Okay, so we look and see. Let's see what we can figure out. A hornless bull is crossed with 3 cows. All right, so it says hornless is dominant and horned is recessive. All right, well, a hornless bull. So we know it's got to be big H something, because that is a dominant trait. And the bull is the male, so he has to give his alleles or gametes to the baby. So let's see. It says cow A is horned. Horned is recessive. So we can kind of figure out cow A's. Cow A is horned, so we know that she must be little h, little h. Okay, their calf, when they have a baby together, is also horned. So it's got to be little h, little h. So in order for that bull to have a horned calf, it must have given it a little h. Because the mom gave it a little h, but the dad also has to give it a little h. So we know the bull must be big H, little h. Okay, we know he's hornless, so he has to have a big H. But because he has a horned calf, then he must be giving it one of a little h. So now we look at corn B. Cow B is also hornless. Okay, so Cow B is hornless, and it has a calf with the bull. So calf B prime is horned. So since this baby is horned, has little h, then cow B, we know it's hornless. It's got to have a big H, but it's got to have a little h to give to that baby. Cow C is also horned. So cow C is horned. It's got to have little h in order to be horned. Their calf, when they make one, is hornless. Okay, so we know it has to have a big H, but since the mom cow has only little h's, it must give a little h to the baby. So it's got to be big H, little h. Okay, so it's kind of a process of deduction. You go through and figure out, okay, well, it's easiest to figure out which ones are little h, figure out which ones are little h, and then work backwards and figure out what the mums and dads had to have. So that was the homework questions that I had assigned for that section. Now we're going to talk about what you do when you have an organism that is showing the dominant trait, but you're not sure whether it's homozygous for that trait, so say big D, or heterozygous, big D, little d. Now, this is kind of important when you're doing breeding or things like that. So when they're looking to breed for a certain trait and they want to know if the father or one of the parents is a carrier of a recessive allele, or if it's got both dominant alleles, then they do what's called a test cross. To do that, they actually mate it with, you take the one that you're not sure about and you mate it with one that you know is homozygous recessive for that trait. And when you do that, you can look at the offspring and compare and figure out, okay, well, the parent that we're not sure about must have this particular genotype. Okay, so here's what we mean. This would be the alleles given by the test individual, the one that you're crossing with, so the homozygous recessive. They have both little alleles. If the one you're wondering about has both big alleles, okay, then it would give a big D and a big D. In that case, this this combination of parents, okay, when they're mated, would end up having all their babies show the dominant trait. They'd all be heterozygous, but they'd all show the dominant trait. There'd be none that show the recessive trait. If instead the unknown parent had the heterozygous or was a carrier of the recessive gene, then because you're mating it with one that's homozygous recessive, some of their children, half of the offspring that are produced, okay, when it's mated with the heterozygous, um, the unknown fellow that's heterozygous, half of the children would show the recessive trait. Okay, so if you were to mate them and find that any of them had the recessive trait, then that would tell you that the unknown parent must be a carrier of that recessive allele. They must be heterozygous, not homozygous dominant. Okay, so this was the example of it that's in your book. Okay, so they're dog breeders, and basically if if the organism was recessive for this particular gene, if they were homozygous recessive, they end up being a dwarf. Okay, a dwarf version of the dog. So in order to breed, a lot of them want to know whether their particular dog, if it's the tall version, is it a carrier of the dwarf allele? In other words, do they have risk of having dwarf babies, or is it one that has both dominant alleles? Okay, so in order to figure out which this particular dog is, they would purposefully mate that dog with a dwarf dog, okay, one that's homozygous recessive, and they look at the babies. If all of the babies are tall, then they'd know that that parent must have been big D, big D. Okay, this is, you can look at what's expected. Okay, we said, like we said earlier, okay, if it was a carrier of the recessive allele, the little d, then they'd expect to see half of the offspring be dwarf. If they have any that are dwarf, then that's a sign that that particular parent that they're wondering about has to be a carrier of that allele. So when they're wondering, they do what's called a test cross. You need to understand what a test cross is. Okay, so this was a question about test cross, I believe. Oh, I need the screen. So, number 15. In mice, brown eyes, big B, are dominant over blue eyes, little b. In your lab, you perform a test cross by mating one brown-eyed male. So if he's brown-eyed, he could be big B, or big B, big B, or big B, little b. And you're mating them with a recessive, one that's recessive for both. Okay, so the female is little b, little b. So you're really performing a test cross to find out whether that male is big B, or big B, little b. They end up with a litter of 8 brown-eyed offspring. So, what would you conclude about the genotype of the male mouse? Well, you would assume he must be big B, because all of the offspring are brown-eyed. Can you be certain beyond? Your lab partner says not so fast. The test cross could just be a matter of chance. So, let's use the product rule to determine the possibility that you might be wrong. Okay, so in other words, we're going to use the product rule and figure out, okay, what is the chances of all 8 being brown if indeed it was a carrier of the recessive trait? Okay, so if the parent was big B, little b instead of big B, then we expect half of the children to be brown-eyed and half of the children to be blue-eyed. So the chances of it being brown-eyed is going to be 1/2, or 2 out of 4, right? So if you have 8 children, or 8 baby mice, then the chances of all of them being brown would be the chance of the first one being brown, 1/2, by the chance of the second one being brown, 1/2, 8 times, okay? So that we get all 8 children. So on the top, 1 by 1 by 1 all that times is 1. 2 times 2 times 2 by itself 8 times is 256. So the likelihood of that happening is 1 in 256 times. It is possible, but not the most likely. Okay, so it is possible. Well, this was just showing again. I got them to do the test crosses to see what the likelihood was. And this is how we got, well, half would be blue and half would be brown, if indeed this guy was heterozygous. Okay, so it is possible that having 8 that are brown-eyed is due to chance, but it's only 1 in 256 probability. Okay, so there's a good chance that that male mouse is big B, but it would be better if you did some more matings to confirm your results. All right, so I think I'll stop and cover the labs separate. Okay, so we'll stop the recording there.